do want to take the time to reintroduce Dr. Oscar Segurado. For those that missed the introduction earlier, he's our chairperson for today, a good friend of our group. He's currently the Chief Medical Officer of ASC Therapeutics. He'll give you guys more information about his wonderful group that is doing amazing things for genes and cells. Currently, he is a CMO, as I mentioned. Before this, he was a CMO of SimVivo, Mirad Genetics, and Cellmax Life. Prior to that, he was also the VP of Becton Dickinson and the global medical head for Abbott AbbVie. He's the author and co-author of 100 peer-reviewed publications, including Nature Magazine, Lancet, and Journal of Experimental Medicine, and many other books and medical articles that you guys can even look up on your own. You can just Google his name and you'll see a long list of articles that he's been a part of. He has his PhD from the University of Würzburg, Germany, and an MD from the University of Salamanca. So without any further ado, I'd like to introduce once again Dr.
Thank you so much. Let me make sure that I get to my presentation. Can you guys see it? Hold on. Let me make sure.
Yeah, it's open.
Okay. Full screen, I hope. So, thank you. I'd like to share with you my perspective on the unique personalized approach required for clinical gene and cell therapy. I had the opportunity to learn clinical development of biologics in the 2000s while working with the Humira team at AbbVie. At that time, anti-TNF, an immunomodulatory cytokine, Humira, was a blockbuster with no need for selecting or monitoring patients. Through my work over the last decade, I switched into gene and cell therapy programs, especially in rare diseases, and I realized how critical it is to apply multiomics and big data analytics in this space. I'm going to discuss two approaches to biomarkers or companion diagnostics that can be applied to personalized gene and cell therapy in clinical trials. First, those biomarkers that are used for patient selection. So we select patients who may benefit from a targeted gene or cell therapy, and then also to monitor patient response, and over time to optimize the risks and benefits of these therapies. But for that, I'm going to use two case studies based on what I'm doing right now. First, we're going to speak about gene therapy, and you're going to see that I'm going to take you into a journey from the lab to the hepatocyte, and you're going to see what I mean by that in a moment. The case study is going to be hemophilia A. And the second case study is from our work in stem cells. By the way, when you hear ASC Therapeutics, ASC means applied stem cells. So that's the background that we have as a company that started working in 2008 on stem cells, and also developed a gene editing technology in collaboration with Stanford University. And through that, we went into this specific indication that I'm going to be discussing with you, which is graft versus host disease. And you're going to learn a little bit about that. So during all these 12 years, we have been ensuring that from being a lab and a service provider for biopharmaceutical companies producing cell lines and so on, now we are able to ourselves ensure that we can develop clinical treatments for very, very serious diseases. So I'm going to start with this slide that was presented on Monday by Peter Marks, who is the director of the CBER, or Center for Biologic Evaluations and Research at the FDA. So he was the meeting I was attending, the virtual meeting, was gene therapies in rare diseases. And he used this analogy that you can see to the right to be able to tell us what's the difference between personalized medicine versus individualized medicine. Personalized medicine has been around for a long time. It's basically ensuring that we give the right drug to the right patient. But these drugs are already produced under the what I would call blockbuster model. What that means is that the pharmaceutical industry has been developing amazing drugs. Again, I had the opportunity to work with Humira that in reality apply and they're going to be very helpful for the majority of patients, but not for all. But there is really not a lot of ways to adapt that medical treatment to different types of patients. So we have a number of indications. In this case, it's mostly a number of autoimmune diseases, rheumatoid arthritis, psoriasis, or Crohn's disease. And this is, as it says here, ready to wear. So practically, it's given to a number of patients with that indication, with that disease, the way it's classified usually under clinical evaluation. And the majority of those patients are going to respond. Some patients are not going to respond. And we know that also from clinical trials. There are a number of patients that respond, others that do not. And we compare with placebo, and if the results are statistically significant, the drug is approved by the FDA. Now, we're moving into a completely different approach, which is individualized medicine, where we are creating the right drug to treat certain patients. These are made to measure, as you can see here. And the reason we are doing this is because there are a number of patients that have diseases that are so-called rare diseases, and the majority of them are monogenic diseases. What that means is that there is a single gene that has either a mutation or there is a full other type of defects on that gene, and we need in some way to replace that gene. We know nowadays of about 5,000 monogenic diseases, and there are now hundreds of companies working in gene therapies for these diseases. What you can see here is that what we heard from Peter Mark this week is that the FDA is committed to advancing the development of cell and gene therapies for populations of all sizes. And this is tremendously relevant because what we are seeing is that they are also helping not only the big guys, the big companies, but the small organizations like ours, working to overcome limitations in manufacturing, which is tremendously complex in the gene therapy space. They are helping us to individualize product development, provide input through a number of mechanisms that he presented, and encourage innovative clinical trial designs. So all this is what is becoming a real shift in the way clinical trials are conducted and the way product and drug development is taking place. And this is the general model of personalized medicine, which is also at some point a tailored approach to improve treatment outcomes. And the key on how to do this is through biomarkers. And when we think about biomarkers, we usually think about those that are measured in the blood, but some of them can also be imaging, can be other type of diagnostics that are going to be help us to identify these green individuals, which are exactly the individuals that are going to benefit from this drug, or those in gray, which are potentially going to benefit, and those that are absolutely not going to be responding to that drug. We want to know that. That's why we're working on a number of organizations on biomarker categories, which are related to safety, diagnostics, susceptibility and risk, prognostics, prediction, response, and monitoring. So all these is the ecosystem that is around personalized medicine. Now, when we go in specifically in gene and cell therapy clinical trials, we are in a little bit of a different situation. First of all, we are now using advanced biologic therapies that possess the intrinsic benefit of being precise tools for manipulating human biology. As I mentioned a moment ago, we already know which gene has a defect, is not working, is not producing a protein, and we are able, because we know this clear pathophysiology, to alter and to make changes. Sometimes you don't need to replace that actual gene that is missing. You can activate another gene that is going to have a similar function. So all this is absolutely relevant, because now what it tells us is that we need, first of all, be able to select patients, which sometimes maybe also correspond totally with the clinical diagnosis. So the patient has a clinical diagnosis, which is hemophilia A, and the majority of these patients are going to have in any kind of inherited defect on factor VIII, which is the one that is required for these patients to have a perfectly functioning clotting system. So we need to genotype target genes or phenotype certain target cells. So we're going to understand that that there are some real targets that we can address with either gene or cell therapy. There are also ways to understand what is happening with these patients, looking into certain of these cells and understanding the functional effects that our therapy is going to have in relevant cells of this individual. Usually this is for blood type of cells. So another important aspect, once we start, especially in the clinical trial space, once we start treating these patients, we want to monitor the patient response over time to assure the maximal patient benefit. And we want to ensure that we understand what are those biomarkers that are going to tell us that besides having all ways to define clinical efficacy, which are usually a number of well-defined parameters, we also want to have some biomarkers that, by the way, the FDA loves them. So they want to have the so-called surrogate biomarkers, which are going to tell us how the biology is working in this precise individual. And obviously, extraordinarily important for all these patients is safety. So we want to make sure that there are a number of safety biomarkers that are going to tell us about the potential of adverse events. Now, let's start with the first case study, which is hemophilia A. This is a disease that is really serious for patients, because they are patients where the blood is not able to clot because there is a lack of one of the factors in the cascade of proteins that are required for clotting the blood, even for a very small wound. The majority of the patients have the so-called hemophilia A, which is the lack of clotting factor VIII. And then there is also hemophilia B, is the lack of clotting factor IX. I'm going to focus on hemophilia A, which is our first target. But indeed, we are also working on hemophilia B and looking into replacing factor IX. And very important here in terms of from the biomarkers perspective is that many of these patients have already, from the outset, a number of antibodies that are going to block the effect of factor VIII. Even if you replace this factor VIII through gene therapy, these patients are going to destroy it through the immune system. So it's important to understand that there are a number of patients, about 15 to 25%, that cannot receive the actual factor VIII. And there are other therapies for these patients, which are a bridge. I'm talking about factor VIII. There is a bridge, let's say, to factor VII to factor X, and you don't need to have this factor VIII. So it's extraordinarily relevant to be able to understand the entire biology to see how we can target these patients. And we have here the situation of, although this is a rare disease, we have about 400,000 people worldwide with hemophilia, and many of them are really diagnosed with A or B. Now, in our organization, what we did, and you're going to see in a moment, we have a a clinical development program that is already leveraging with work that was done by the Emory University and it's a company called Expression Therapeutics, where they developed a specific factor VIII and a combination with what we are going to see in a moment, what I mean by promoter and so on, and a capsid, where we are going to infuse this construct into a patient, and the patient is going to have, with just one infusion, they're going to be able to practically keep going the rest of their lives, if everything goes the right way, without any kind of replacement therapy for a factor VIII, because factor VIII is going to be produced by their own body. And I'm going to use, when I was talking about lab to hepatocyte, I'm going to use an analogy which is what happened with many years ago with the Apollo 11 journey from the Earth to the Moon. And I'm going to explain you how, in a very, I think, a very useful, easy way, how gene therapy works. And for that, we're going to talk about the actual rocket. We're going to talk about the capsule and the lunar module, and how the lunar module either stays around orbiting the moon or lands in the moon. And now, by the way, we have to be careful. We have to talk about the rovers landing in Mars. So, first of all, we are working with an adeno-associated virus. It's called AAV8, and this is the rocket. This is the rocket that really incorporates all the information that we are going to give to the hepatocyte, in this case, to be able to integrate this information and start producing factor VIII in our case. What happens with this adeno-associated virus is that it's totally harmless. So it's a virus that, by the way, is used also for some of the vaccines, Johnson & Johnson and AstraZeneca, and many of the ways we are using these platforms are also being used by RNA and DNA vaccines. So we are sometimes competing for manufacturing capacity, believe it or not, and some of the providers, CDMOs or that help us manufacture our viruses are really squeezed right now. So this is it's a little bit complicated, the slide, but I'm going to try to help you go through it. And this is, again, we're now talking about the capsule and the lunar module. Imagine that the capsule is what we call a promoter. It's the one that takes the all information and makes sure that the lunar module orbits or lands in the right place. And what is different about our approach is that we're using a transgene that is a recombinant of human and porcine. Why are we doing this? Because we are now producing, in contrast to a number of companies that are working on exactly the same approach, we have a second-generation gene therapy. Why is that? Because this group in Emory that I mentioned a moment ago had been working for a long time on looking into how to increase the biosynthesis and secretion of factor VIII. And you can see to the right how there are two main processes that take place in the nucleus. First, there is the transcription, where the DNA is converted into RNA, and the RNA is converted into a protein. All this process is improved because the way we're using a promoter and the transgene. Again, the actual promoter or the capsule and the lunar module. And we conducted a number of studies that showed that when we put this combination, this promoter and transgene, circulating around or orbiting, or let's call it that way, orbiting around the nucleus, it is going to produce factor VIII protein levels. I am showing you here preclinical study work, but this is what is going to lead us into a clinical study, a phase 1 clinical study that is about to start in the next few months when we get IND clearance from the agency. And what we also are developing, because it's also in our DNA, let's call it the way we work, is being able to also land that lunar module in the nucleus and then being integrated into the genome of the individual. That is not what happened with the previous example that I gave you. So we're not really integrating this sequence of factor VIII into the genome or in the nucleus. In this case, we're using hepatocyte, a liver cell. But we have experienced this paper down there from in Nature was created by our team, and we are using also an approach where at this point is still preclinical. We are about to approach the agency, the FDA, to show our work and how we are able to do that. The advantage of doing this is that the moment that is integrated into the genome of the individual, even if the hepatocyte replicates, is still going to be able to keep producing factor VIII. And very important here is the mission control, what you can see here, which is, again, and I'm not going to go into the details because this is specific to our case study, but we identify patients with severe hemophilia. We have a number of selection biomarkers. So we monitor, we have an absolute control of what patients are going to enter our clinical trials. We look into the existence of these antibodies against the factor VIII protein, potentially some antibodies to our capsid, to our virus, AAV8, then a number of liver function tests. And once the patients enter into the study, we're going to monitor those patients through a number of a period of time. Usually the first year is the most intensive follow-up, where we look into especially safety measures. We want to make sure that these viruses are actually infecting the liver cells. We are excluding other organs and tissues. We look also into the potential generation of new antibodies to these viral capsids, and we look into how the immune response is taking place in these individuals. And the most important one is at the end, this efficacy, factor VIII activity. We measure in an individual that doesn't have any production of factor VIII, how this factor VIII is active. And there are a number of tests that tell us ex vivo how this patient is doing. So that's it in terms of gene therapy. And for cell therapy, I'm going to run very quickly about our project, which is focused on patients that have something called graft versus host disease. And this is patients that have a bone marrow transplantation. What it means is that they receive stem cells from another individual. And as you can imagine, when that happens, it's very possible that these cells, although we try to ensure that there is as much of a match in terms of HLA from the donor and the recipient, there are many situations where this is what happens. And about 30 to 70% of the patients develop this acute graft versus host disease that sometimes recovers automatically or briefly. Sometimes they can even go into a chronic graft versus host disease. So this is a very serious condition. It can lead to death. There is an unmet need of being able to treat these patients. What we did is that we went to the one of the top centers in the world, is the Karolinska Institute in Stockholm, and we in-licensed from them a technology which takes advantage of the placenta. The placenta is also called the decidua because it's like in certain trees that is practically discarded. But there are certain cells there that do tremendous work protecting the fetus from the immune system of the mother that potentially could attack the fetus because it has certain, obviously, 50% of their cells come from the father. So we had a very thorough tech transfer. We were able to take those cells. We were able to isolate them, expand them, and we characterize those cells, and realize that they indeed protect the fetus from the mother because they have a number of immune characteristics. I'm not going to go into these immunology type of details, but at the end of the day, they control acute inflammation. They reduce the effect of what are called autoreactive cytotoxic T cells and increase other cells. So at the end of the day, the overall response rate in our preliminary work in about 50 patients is close to 80% success. So in these cases, again, we have this a very defined population. We look into ways to select those patients that are going to be most likely responding to this therapy. We do immune profiling of the circulating T cells and the cytokines of these individuals. In other words, we are looking into their immune system. We also look into how certain cells, which are specifically in the gut and the skin, which are the target organs from this graft versus host disease, take place. And we also look into effects of other therapies that are used and how these patients are responding to them. And again, we also follow these patients. This is an acute disease, so as you can see here, is more about just looking into the first month or first 2 months. And we want to make sure that we understand safety parameters. And there are a number of predictors of immune-related adverse events. We also do immune profiling with different ways, and we phenotype our cells, so we are able to understand how they are doing in each individual patient. Again, the points, and thank you so much for your time. The point I've been making here is that we are really moving into a different world and a different way of looking into diseases, looking into therapies, and combining this individualized approach, which goes even beyond the personalized medicine structure that we have been going on for many years. Thank you so much.
Thank you so much, Dr. Oscar, for your amazing presentation. And before we move on to the next presentation, if there's any questions for Dr. Oscar, you can write it in the Q&A tab to the right or in the Discuss tab as well. I can see a couple thumbs up have been coming up for Dr. Oscar, which is great. A couple hands have been raised as well from Marietta. So Marietta, do you have a question for Dr. Oscar? I can see that your hand is up. I saw another couple thumbs up are coming up for you, which is great. We are still a little early for the next session, so yeah, we'll take our time. I know a lot of people in the room loved your talk based on the thumbs up that we're getting on our end, my screen. So.
Yeah, maybe I can just add a few points.
Yeah, Dennis Meir, I believe VP ClinOps asked you a question.
Okay.
He said AVV5 versus AVV8.
Wow. I don't think I'm the right person to answer that, but what I can tell you is that there are differences on the type of capsids or viruses that we're using. And every one of us in our industry is trying to focus on one platform. And in our case, it's AV8. Why is that? And I'm sorry, I don't know the details on AV5, but I can tell you that there is, I think it's a Spark and Roche that are using AV5 or is Biomarine, also with the same target, the patients with hemophilia A and the factor VIII. So it can be used. So you can have different type of vectors addressing different types of diseases. But in our case, the reason we selected AAV8 is because it has a better tropism for the liver. And I think that that's something that makes it interesting. Now, I worked before joining ASC Therapeutics. I was working with Avexis, which was acquired by Novartis, and their platform is AAV9. Why AAV9? So AAV9 is because it has a better tropism for neural cells, nervous cells, especially for motor neurons. And the motor neuron is where you want to produce the SMN protein that is required for patients with the spinal muscular atrophy. And this has been one of the best successes in the industry. There are many patients that have been treated with these type of therapies. And the most recent one patient even make it to the news, and I think it was also this week probably, is a 4-day-old child that had already have been diagnosed with spinal muscular atrophy. Let me just highlight here how these patients, until Zolgensma, which is this therapy, was available, would die within 1 year. And we have now patients that are 5 and between 5 and 10 years old. I don't know exactly how old they are, but this is an example of what we can achieve with this kind of therapies. Again, I think AAV5 is also available and useful for hemophilia and also for factor VIII replacement. And factor VIII, AAV8 is the one that we like in and where we are experienced. And you have another example, AAV9, for other type of patients that have different type of targets. So it depends on the cell that you're targeting and how you are going to be able to construct your combination of remember the lunar module, the capsule, and the rocket. So all together they do the right thing. They go to the right target tissue and organ.
Wonderful. Is there any other question for Dr. Oscar in the room? I saw a few more thumbs up that came in, which is great. Keep the thumbs up coming. And we will take one more. Otherwise, everyone then can go to the next room, which would be Accelerating Clinical Trials, Innovative Design. So everyone can head over to the next session. I would appreciate it.